Get some BME faculty to be present.
Yes.
Yeah, Carly.
I'm not on the committee.
Oh, you're not? Me, Rob, and who? Do I count as faculty?
I don't think I'm on the committee.
Once upon a time,
Luke counts. Okay. And so they got it on the terminal over there, like on the screen, hit the pound sign, number sign.
wait, but just say
That's good.
seconds.
Thanks, Robbie.
All right. Good morning, everybody. Why don't we get started? Welcome to Bram's PhD defense. The logistics of it would be he'll present for about an hour with questions by everybody at the end of the hour. And then we'll ask the rest of the people, meaning non-committee members, to leave. We'll get an answer by the committee. Then we'll have some our internal deliberations and an outcome to be decided based on Bram's presentation, questions that he answers, and all this sort of stuff. Before we get there, Bram came and started my lab with a degree in chemical engineering. So it's a little bit of not the most traditional path for a BME student. It took him a little bit of time to get up to speed and has done some great work in AFib. We have been doing lots of studies looking at EGMs and whatnot, and has led to Bram being a finalist at Heart Rhythm Society, which is a big deal. He's gotten 2 papers published in good journals already. He'll obviously tell you a lot more about it. So without further ado, Bram, all yours.
Thank you, Robbie. The heart is responsible for pumping blood through the rest of the body, and it is composed of 4 chambers. The bottom 2 are the ventricles, and the top 2 are the atria. Let's see. Now, the heart is made of about 10 billion cells, 3 billion of which are responsible for the contraction. But they don't just operate all at once without outside input. They are electrically coordinated. Here is a diagram of how electrical impulses spread through the heart, going first through the atria, then through the ventricles. And shortly thereafter, the muscle cells contract. I liken this process to taking a rock and dropping it into a pond. And there is a clean ripple that reaches every aspect of the pond. Now, this is not always the case. For some people, instead of having a clean pumping motion, their atria, they quiver. This is an electrical problem of the heart. And as we can see in this figure, it's like there's millions of little eddies, chaos. And in fact, in the first work looking at this, they described it as a vermiform movement of the auricles, vermiform meaning worm-like. And it's sort of equivalent to if you took a big bucket of gravel and tossed it in the pond instead. So the clean ripple, we have chaos. This is atrial fibrillation, which is the subject of my dissertation defense today. I am Bram Hunt. I am a doctoral candidate in the lab of Robbie Ranjan here at the University of Utah. Our lab studies arrhythmias, and over the last 6 years, I have focused my research on atrial fibrillation. I'd like to give an acknowledgement to my committee for being here, and thank you for their mentorship. I could not have done this without the support of many people. Now let's talk a little bit more about atrial fibrillation and why it matters. First off, it affects about 60 million people around the globe. That is expected to increase dramatically over the next few decades as the population of the Earth ages. And your lifetime risk of getting it is about 25%. This is then a problem because it increases your risk of stroke by around 5-fold. And then similarly, it increases your all-cause chance of dying by about 2-fold. And what's more, our methods for treating it are mixed in efficacy. And this is due to a number of things, but one of the aspects is that the exact cause and the mechanisms sustaining it aren't clear. Now I'm going to talk about the proposed reason for atrial fibrillation This is the idea of drivers. Drivers are discrete sites in the heart sending out simply bad electrical signal. And this interferes with the heart's normal electrical function. They come in two main forms. The first is a focal ectopy, an ectopic beat, where from one site we see like another ripple being dropped, competing with the heart's normal ripple. The second is because of the nature of the heart's electrical conduction, you can have the ripples re-enter each other and enter a self-perpetuating rotor. Now, these have been the subject of study for some time, but clinical results on these are mixed. And trying to burn them to help cure atrial fibrillation. Now, for this other aspect I want to talk about, let's consider conduction. Here are two cardiomyocytes. Those are the main muscle cells of the heart. And the electrical propagations spread from one cell to the other based on their connections. This is conduction. And when you have a whole lot of cells connected together, it becomes a wave. When the heart is damaged, or in the case of atrial fibrillation, sometimes you will have regions become non-conductive, and this disrupts the propagation of electricity through the heart. This is thought to be involved in atrial fibrillation, and generally we will call this fibrosis. And it would be nice to incorporate this more into our clinical methods, into our research, but it ends up being very difficult to study. The gold standard is to slice the heart and then to go investigate the cells under a microscope, but that is, of course, impractical for patients. This leads us to our gaps in literature that we hope to address today. The first is that even though that some studies have seen drivers in the atria, they haven't been investigated over a long time. We don't know if they stick around from 1 month to the other. And the question then becomes, if they only stick around for a short period of time, is it really worth treating them? The second is that another issue with drivers is that we don't have a gold standard automated method for finding them. So it's slow, and even though it would be a task well suited to machine learning, as we will see in a bit, our machine learning approaches are limited by small data sets. And third, as we were discussing, fibrosis may be relevant to the AFib mechanism, but it's difficult to study. And using our current method, MRI, for detecting it is inconvenient and expensive. Now let us look at our research aims for today. The first is I want to identify the locations of drivers over serial mapping studies, looking how they progress with time. The second is to develop improved machine learning classifiers using a method of getting around small data set size, pre-training. And third was to develop a method for finding fibrosis using electrograms, allowing physicians to potentially find fibrosis without needing MRI or histology. Looking at aim 1. Let us discuss, this is the main source of data for this defense. We have a preclinical model, canine model of atrial fibrillation, where we implant a pacemaker into the right atrium. And we set it to pace very fast, 1 second on, 1 second off. And about 2 to 6 weeks later, they will sustain atrial fibrillation without even pacing, at which point we downgrade the pacing to a minimal level. 1 month later, after sustaining AFib for more than 20 minutes, we go and bring them into the EP lab and do intense electrical investigation of the heart. 3 months later, we do that again. And 6 months later, we do that again. In all these studies, we use the Arrhythmia mapping system and the Orion catheter. This is an ultra high density electrogram capturing system. Electrograms are the electrical recordings from the heart. To get more in detail about how we did this, here's an example, left atrium, and inside it, a catheter. This is one site, and we would leave the catheter there and capture electrograms for about 3 minutes. And then we would move it around and then capture 3 minutes of electrograms at each of these sites such that we had a really high density mapping. Then from these, we looked at the fastest 2 seconds of EGMs and went to find drivers manually. We developed a software for doing this, which I built to go and display the catheter position in the atrium with real-time playback of the electrograms, with a splayed out spline view to help us look at veins, to have interactive controls for handling the electrocardiograms and the electrograms to see what the data quality really looked like. And then finally, a set of filters that we customized to our driver detection task. Let's look at an example of finding a driver. Here I've zoomed in on the 3D mapping in the GUI we built. And we'll see yellow impulses, assuming we can look at this animation well, spreading throughout. And those are the electrical impulses. And we see a sort of clockwise motion. This might be an example of a rotor. And we looked at about 800 sites to find drivers or not. So what did we see? Well, our first major insight was that the number of drivers increased from an average of about 5 to 7 over 6 months of atrial fibrillation, or 5 months here. This is a really good indicator that we need to be treating patients early, as fast as we can, to prevent the atria from developing further in their atrial fibrillation. And this has sort of been seen in other studies, but this is the first study to look at serial mapping in the same subjects over time. So then we were considering, okay, that's great, the number increases over time, but how do specific regions develop? Maybe there's some interesting insights there. To do this, let's look at an example. Here's the left atrium from the endocardial surface inside the heart, and this is the right atrium. We took these and we broke them up into about 15 major anatomical sections and looked for the presence or absence of drivers in those sections over time. Here's an example of the left superior pulmonary vein, where we saw in green, no driver, red, a driver. From 1, 3, and 6 months. And then we looked at all subjects to see how at that particular site it was changing with time. The first thing we'll note is there are some cases where no driver, driver, no driver. These are unstable locations. You probably don't want to hit these if you are a clinician. Second is sometimes we'd see a driver and it would show up at the following study, but not necessarily all the time. That gives more credence to the fact that there may be reason to hit those. There may be more of a basis for them existing. And finally, we delineated this type of driver we're calling multi-recurring, where the driver showed up in the beginning and at every subsequent study. These likely have the most basis to their treatment. We next were curious, where do we see these multi-recurring drivers? Well, the first thing that we note is that the left atrium had the vast majority of them, about 30 out of 31. But beyond that, even the left atrial appendage had a lot of multi-recurring drivers, but there was no stereotype. This leads us to a conclusion here that you want to tailor your AFib ablation to the patient you're treating because there's no general anatomical rules that you can just follow to hit these. Now, we can't go and take a patient and measure their AFib over 6 months to find these things. So we'd be curious about, are there markers of these early on that we could use to help find them? I'm going to talk about cycle length, which is roughly how slow a driver is activating. And what we saw was that non-drivers were activating slower than non-recurring drivers, which activated slower than recurring drivers. This promises that there are some electrical features that we can use to find multi-recurring drivers. And again, we looked at recurring versus multi-recurring, and we saw that multi-recurring is the fastest. So there may be some feature we could use. We then became curious about another marker. Maybe we can go look at fibrosis with MRI to maybe find these things. So this is with some help of Eugene Kwan, who was responsible for much of the MRI work, where we go and here's an MRI slice, and we've segmented out the left atrial wall and the blood pool, and then flesh that out into a map of fibrosis and nominally healthy tissue, here with green being fibrosed, blue being healthy. And we associated that with our drivers. The first insight was that drivers were associated in general with fibrosis. That means that in general, there may be some sort of fibrotic basis for those drivers. However, we didn't see any difference between non-drivers, non-recurring, and any recurring. So it's not a good predictor of recurrence of these drivers. Now, in applying this information we've gathered to clinic, we need to be careful. First, here we have a preclinical model with a common pathogenesis of AFib. They all have this rapid stimulation. Even though they maintain AFib over time without the pacing, it all came from the same reason. So we'd have to go look at these in the clinic to really have an idea of whether this generalizes. Next, we only looked at the fastest 2 seconds from each site to find drivers. In this way, the number of drivers, it might be underestimated. There may be a lot more than we thought. And we also consider that the basis of recurrence here is still unclear. To really understand this, there is a need for histology, genetic analysis, something to help us figure out what's happening at these sites. And again, to reiterate, we don't want each patient to have 6 months of study before we can treat them. So a single study detection method would be needed before we can move this to the clinic. So what we conclude over here, drivers increase in prominence over time. This supports early intervention, fast as you can. Second is that fibrosis and drivers are linked, which gives some evidence that maybe there is something there in the tissue that helps them sustain, and maybe they're real. We found here for the first time evidence of stable drivers over multiple months, and we identified some major, like these 2 major categories, recurring and multi-recurring, but there's no stereotypical location for these recurring drivers. And that requires patient-specific treatment. This work is now published in Europace and was the subject of me being a finalist for the Young Investigator Award at the Heart Rhythm Society conference, which is I would say, the foremost international conference on cardiac electrophysiology. Let's now move to aim 2, the machine learning aim, where again, what we want to do is try to find drivers with machine learning using pre-training to get around the small dataset size limit. We take a little bit of water. Let's work through how this goes, the deep learning detection. Here's the left atrium. We move the Orion catheter in there to collect electrograms. Again, from aim 1, we feed this into a black box, which I will explain a little bit, a deep neural network, and this will send out whether at that site there is a driver or not. Let's work a little bit on how classifiers work. This will help us understand the nature of the work that we've done here. So classifiers in general are just a function that maps an input to a class. You give it something, it says yes or no, or which class it is. A very basic example would be at the amusement park. Maybe you have a "you must be this tall to ride" sign, where the input is your height. The output is whether you're allowed or not allowed. Very simple classifier, right? Pretty much all classification works like this. Let's make this a little more complex, looking at multiple dimensions. Here's a picture of a banana. Let's simplify that, because that's a lot, into only 2 pixels. What we can do with these 2 pixels is go project them onto a grid with intensity of pixel 1, intensity of pixel 2, and the banana will occupy some space there, right? This is what we will call pixel space. So suppose I have bananas, and I want to distinguish them from pomegranates. I could potentially go and plot out all the bananas on pixel space, all the pomegranates on pixel space. And in the best case scenario, it looks like this. And you can easily see how, you could just draw a line, y equals mx plus b. That's 2 parameters to distinguish between pictures of bananas and pomegranates. That's a fortunate case, though. Usually with real data, it looks something like this. Pictures of bananas, pictures of pomegranates. You can see evidently a single line is not going to work there for distinguishing them. And you're going to have a more sophisticated function to wrap around one class. Notably, this is going to require a lot of parameters because this is a complex equation. This is not y equals mx plus b. This is many, many more parameters. And one of the aspects of machine learning is that the more parameters in your model there are, the more data you need to train it. But what if there was a way to go and take data that looked like that, real data, and make it simpler? Bring it more to this case where we have 2 easily distinguishable classes. We're going to approach that with a method called SimClear. I guess what I wanted to note here is that this would take only 2 parameters to train on this, much simpler than our previous model. And this is what we would call a more effective representation. This is representation learning. Now let's look how we would use pre-training on unlabeled data to help us in this process. So here are some bananas and pomegranates, and maybe we have a bunch of random images scattered within our data set. We don't know whether they're bananas, pomegranates, or some other fruit. SimClear, as I was saying, is a method of using those unlabeled data to transform them into better representation. The way it works is, here is one datum. Let's say it's an orange. We're going to color it, and that's because we're going to make it into its own class. And we're going to take it as a mother datum to create a bunch of subdata from augmentations of that orange. For example, we might go and crop that orange. We might do run an edge detection on the orange. Or maybe put a Gaussian blur or add Gaussian noise. So what we see here is that each unlabeled datum now becomes its own class, where all these are related to each other because they come from the same mother image. We're approaching our full data set of unlabeled data. Each becomes its own class. We take one green datum, augment it again. And then we do that for every single element of the data. And then we learn a transform to go and map them together. So we train the network to go and move pink to pink, green to green, blue to blue. Everything from the same mother image gets mushed together because they're responding to the augmentations in the same way. And how we apply this to the labeled data is like this. This is the whole process. We take the unlabeled data, make them into their own classes, augment them, cluster them, and then we add in the labeled data, run it through that network, and in the ideal case, what happens is it will cluster things with similar properties together. In theory, the things that you wanted to discriminate between in the first place. And then here you could easily see from our complex data, we can now draw a clean line between the classes. Let's approach our real data set now. Here is the Orion catheter. It has 64 electrodes. Each electrode captures one electrogram at a time. Here's an example of the deflections during atrial fibrillation. We can go and stack together all the electrograms from all the electrodes. And then we want to treat this like an image. So what we're going to do is color it according to the height, according to the voltage values. You can see that on the screen. And then we can stack it together. What we get is something that looks like an image. And this is where we can use this SimClear process, which is 4 images on our electrograms. To describe our data set, we have about 709 labeled pictures here. When we apply our augmentations, we use essentially the same augmentations I talked about before, like random cropping, edge detection, blurring, and just use a mother image to make daughter images, as we showed before. For those who are interested in the more technical aspects of this, here's our layout of this whole process. We have this pre-training where we go and we have some number of samples on a batch. We feed them through random augmentations. Then we train a 3D ResNet to project them into new representations. Then use this NT-Zent loss to make it put those representations together. We trained this on 130,000 electrograms. Then we ripped out that 3D ResNet and put it into the fine-tuning task. So we put on a new linear classifier at the end, and we just feed in our labeled samples of electrograms, and it decides is that a driver or not a driver. We did this with about 600 electrograms. What we found was pretty strong results, where we saw our pre-trained network increase by about 20% accuracy over the non-pre-trained network, a significant improvement, with F1 scores being similar. For those who don't know, F1 is an average of precision and recall, with our data set of about 110. Now, we weren't just satisfied with this. We were curious, what is making the network tick? It's a black box as is, but hopefully we can find something from it that gives us some idea of what it's doing. Is it making sensible decisions, or is it memorizing the test's answers? So we use this thing called GradCam. Here are some images of cars. And if we were to train a classifier to find cars in an image, we would hope it was paying attention to the cars. So there's this process we use, GradCam, to go and create these heat maps of what was important to the attention of the network. And we see here in our classifier example that it is paying attention to the cars to make its decisions, which is good. It probably shouldn't be paying attention to the graphs to decide if there's a car there or not, right? We then applied this to our network, where we saw reasonable attention. Here I have one of those electrogram images colored from purple to yellow based on the importance to the network of the various regions inside the electrograms. In between these two red lines is where I manually said there was a driver here in these sets of electrograms, right? And we see that it put a nice fat line when it classified this as a driver in that section. That's very promising. And what's more, when I went to look at this after the fact, and I looked at this section where it was paying a lot of attention to this, this looked like a driver on a second pass. So that is evidence of some reasonable attention. And additionally, when we went and looked at the true negatives, the attention was diffuse and sparse. That's good evidence that it's making good decisions. We weren't just happy with this. Part of the idea of this whole problem was that this pre-training allows us to train networks with less data. So we just reduced the fraction of the training driver data set for both pre-trained and non-pre-trained networks. And we saw that the pre-trained network is definitely more resilient than the non-pre-trained. And you can go up to a 30% reduction in the training data while still maintaining an edge over the non-pre-trained data, or the non-pre-trained. We then were curious, okay, that's great and all, but there's a lot of choice in how you might augment your data in the SimClear process. So we investigated a large number of different augmentation types. This is sorted by F1 score. I showed the best, which used rotation, crop, and Gaussian blur. One of the things we note here is rotation is an important augmentation, where when you're in the clinic and you have a catheter inside the heart, it really shouldn't matter what rotation the physician has put on that catheter. So it makes sense that that would be important augmentation in this process. An additional conclusion we can make here is we look at the best pre-trained network and the worst pre-trained network, and there's a huge difference. This shows that you really need to pick your augmentations with care when you do this. And you need to think about not just things that work on image data in machine learning projects that are more theoretical, but you need to consider your domain and appropriate augmentations for that domain, in this case, cardiac electrophysiology. Let's talk about the limitations and what's next for this work. First is that there's other relevant augmentations that are untested. The big one right now being time clipping. It would make sense that if something's a driver, it would probably be a driver in any given section. Second is that our network is pretty small. It only had 18 layers for ResNet. Larger networks have been shown to benefit really well from pre-training. So you may be able to see even bigger benefits with bigger networks here. We also need to look at testing with clinical data. This is trained on a preclinical data set. It's unclear how this will work on patients. And then finally, as with all machine learning problems, you can increase the accuracy the more data you have, generally. So our conclusions here are, one, that SimClear pre-training clearly improved accuracy in driver detection. Two, the pre-trained network was more durable to the smaller data set. And that selection of augmentations, very important to performance. You can't just pick any random ones. This work is now in press at Heart-Rhythm O2. And it is open access if anyone wants to read it. Same with the previous EuroPace paper. Let's now look at our AIM-3. Seeing that it's inconvenient to use MRI and infeasible to do histology and clinical data to find fibrosis, we looked to see, can we use machine learning to find fibrosis from electrograms inside the heart? Now, this has already been sort of attempted with these things called low voltage areas. Here's an example of an electrogram, bipolar electrogram from during sinus rhythm, that is normal heart function. And the way we find low voltage areas is we go look at this deflection. This is when the heart tissue is beginning to activate at a particular site. We look at that magnitude, and if that magnitude is less than 0.5 millivolts, that's generally considered that's generally considered a low voltage area. This has been used pretty frequently to predict scar, dense scar, but it's really not a great predictor of fibrosis. So we sought to improve on this with electrograms during atrial fibrillation. One of the things you'll note is that it's really a lot more dense, a lot more activities happening during atrial fibrillation, and I would call that something that makes it richer. There's more data for a machine learning network to latch onto. Additionally, the atrial fibrillation data will reflect different conduction pathways and not just one from the sinus rhythm. Now, to work out our machine learning problem is very similar to above, where here's the left atrium. We have our catheter inside of it. It captures electrograms, feeds that into a deep neural network, and then we use that to reproduce LG-MRI maps from the endocardial surface. There are a number of different ways to tackle this. We first considered, as with doing something like in our driver analysis, where we look at the fibrosis of an entire anatomical site. Here's the left atrium with our Orion catheter, and maybe we just predict one big section and average there. This is probably very feasible, but it's not going to be the most useful. And additionally, we'd be predicting things that are beyond the extent of the catheter. So we went and considered, how about just looking at the fibrosis near each electrode recording? So in the same way, we just go and find the region around the catheter and then try to build something a little bit more high density than the areas or than the regions before to create maps of fibrosis. But then we considered, how about, let's look at the simplest, stupidest possible way of doing this. Let's just go take the Orion catheter, which has 64 electrodes. Just go take one electrode at a time and try to guess whether it's fibrotic around there or not. And then do that for every electrode all throughout the atrium. An advantage of this is that this would be the highest density maps that you could do. And then additionally, it would be catheter agnostic, because when you train a machine learning network, it tends to be reliant on the catheter you used. So here, by ignoring the spatial components and just looking at 1D electrograms, we can use it with any given catheter. And that's what we ended up picking for this project. To go through how this is again, we take a single electrode from the Orion, map that down to a 2-second electrogram. We feed this into a 1D ResNet. We used a network that was previously used for collagen detection ex vivo, which had shown some promise. And then we used that network to predict whether a given vertex closest to each electrogram was fibrotic or not. And we had about 25,000 electrograms for this. Our finding was that when looking at 3 different subjects in the testing data set, we got accuracies of about 95% and an F1 of about 86. This is really strong, much better than I had expected for the 1D electrogram challenge. So what does that look like? Well, let's look at the ground truth. This is the LGE MRI. Its endocardial surface projected onto our arrhythmia anatomy. Here's a cutout for the mitral valve. We have a back view and a front view of the left atrium. And we see that there is big regions of fibrosis in red up on the roof, around the RSPV, and at the lateral wall. When we go and bring the network in and have it make its predictions, we see actually good correspondence, where it is finding big regions of fibrosis on the roof, RSPV, and the lateral wall. And if you're curious, this white section is where we have something in between fibrosis and not, because we sometimes had more than one electrogram for each vertex. And in those cases, for the sake of mapping this, I took its guess as being an average of all the electrograms. Let's look at the other 2 maps that were made. Top is ground truth, bottom is the model's guess. See good correspondence. Again, roof wall reaching over to the RSPV, something on the lateral wall. And then the other subject, the final one, again, it's predicting clean regions where there's clean regions and finding lots of fibrosis where it was seen in the LGE MRI. Now, this isn't just sufficient by itself. We were curious, how do low voltage areas do on this task? What we found was abysmal results. There were no low voltage areas. Here's the same LG MRI as the first example. And here's our predicted fibrosis from our low voltage areas. There are none, so there is no fibrosis predicted. Now, there are some limitations to this work. For one, this only considers endocardial fibrosis. And the field of atrial fibrillation is increasingly looking at epi and endocardial and transmural conduction as being involved in atrial fibrillation. So it would be much nicer if we could go take this network and try to predict fibrosis on the endo and epicardial surface to more fully replicate the LG MRI. Second, this MRI is not considered a perfect surrogate of fibrosis. So to really strengthen these results, we could do something like histology and try mapping between electrograms and histologically predicted fibrosis. And then finally, the model wasn't used against dense fibrosis or scar. So further evaluation on clinical data would be needed. So in conclusion, we found that maps of MRI-based fibrosis can be predicted with our electrograms during machine learning or using machine learning. And these perform the conventional low voltage area-based maps. And this is also useful in that sometimes when you bring a patient into the clinic, you can't bring them into sinus rhythm. So if you only had AFib data, you could still use this. And then the single electrode network is agnostic to catheter choice, as I said before. And also, I'd say that in general, this advocates for atrial fibrillation mapping, which is not a current clinical standard. The standard is they come in and they ablate you, and mapping isn't necessarily needed. So the manuscript for this is written and it's under co-author review with plans to submit it soon. Now let's reflect on the work that was done and form some overall conclusions. First, with aim 1, we showed that drivers of atrial fibrillation have some sort of stability. About 66% of all drivers recurred at the next study, and we identified that some of them are multi-recurring. They're very stable over a 6-month period or a 5-month period. Particularly, I'll note in the left atrial appendage, posterior wall, and roof. Additionally, for AIM-1, we provided evidence supporting that there is a substrate link to drivers. Drivers have some potentially anchoring or relationship to fibrosis. For AIM-2, we showed that our SimClear process successfully enhanced driver detection, with some notes that you can't just do this process all the time. You need to really carefully pick the augmentations chosen. And for our final conclusion, we developed a machine learning model that can predict fibrosis from atrial fibrillation electrograms. And just to reiterate, our ground truth, our low voltage area prediction, and our model's prediction. We just see that you can't find these things on with just low voltage areas. You need something else. And this leads us to the overall contributions of this dissertation work, which is that we've developed these advanced electrogram-based tools, machine learning algorithms, which can help make more targeted, real-time, personalized strategies for atrial fibrillation treatment. This has produced a lot of work. First, let's look at the journal publications, which I've been on and which I've authored over the last 6 years. In addition to that, there have been numerous conference proceedings papers. I've done numerous seminars, posters, presentations. These two here won first prize podium presentation awards. And this here is my young investigator finalist work. Finally, I'd like to acknowledge my advisory committee again for being here, for mentoring me, for supporting me through this whole process. I'd like to give my great appreciation for the other members of the Ron John Laboratory. This work is impossible for a single person to do on their own, and I could not have done it without them. I'm also thankful to many other people who've been involved in this work. And I'm particularly grateful for my stalwart supporter, Allison, my wife, who is here today. And also my little precious lady dog, who has been a similarly stalwart supporter, though perhaps a little sleepy at times. Finally, I'd like to thank you, the audience, for being here. I'd like to open up the floor to any questions you may have.
Sort of questions for Bram.
I just wanted to first say this was amazing. Like it's so clear. Like it's not much to be addressed. In one of the questions you mentioned about recurring.
Yeah.
So does it mean recurring can actually go off and become like normal?
Can you repeat that? Can recurring go off and
recurring?
Yeah. Yeah.
Multi-recurring means that region, it keeps coming up over a period of time.
Yes.
What about recurring?
Recurring, that means it didn't come up again. It showed up. So it's an intermediate stage between just totally unstable and multi-recurring. So there was a discontinuity in it being there between studies.
Could you say that this region is not normal, but it got better?
That's unclear. That's unclear. There are certainly electrophysiological differences between multi-recurring and recurring drivers. Maybe there are things that can be done to predict whether a site will get better with time or not.
Yes, that's the reason I asked this. So instead of can, what predicts this multi-recurrence? Maybe if you can focus on, or if you can see if there's a difference between multi-recurring versus recurring, and see if this region got, or any voltage difference, or any fibrotic difference, or any substrate difference, essentially, between the recurring, multi-recurring, and normal, maybe they can give an idea.
We did look at that, and we saw that there was no difference in LG predicted fibrosis between recurring and multi-recurring. It was strictly limited to the functional properties, where the multi-recurring had the highest dominant frequency, the lowest cycle length. So it was activating the fastest, faster than the just singly recurring.
Thanks.
Thank you for the question.
Great. Great presentation. I have two questions, which I hate questions, but I'm going to do it. I guess my first question is for you on all of the machine applications and scores. How good is good enough clinically? So like, what is the scores that you're reaching for, and what point would this really become clinically viable? And at what point do these scores that you mentioned actually mean that it's...
Okay, that's a fabulous question. And the answer is that it's unclear. It's sort of when physicians become comfortable enough that they feel this is worth acting on. And then additionally, you'll need to look at how does this work in a large scale clinical trial, right? To see how good is good enough. That isn't clear. I'd say anecdotally, talking with physicians, they're looking for something in the 85% plus accuracy range to really say, okay, that's something where I'm getting comfortable trying that out.
Right. And my second question is on noise and I guess alterations to your input. So whether it's coming from a signal and you may get a lot of noise in your data, and also in terms of detecting fibrosis from LG-MRI using different threshold cutoffs for said fibrosis, how does that influence the output and the ability to train, have a successful network in your applications here?
The noise is very much a difficult problem when it comes to atrial fibrillation electrograms. I have an extensive process that I go through with filtering and QRS subtraction and whatnot to go and make driver detection at least something that I found reliable. We did reliability studies to go see, I don't know if I have that right here, like how we're doing on getting rid of noise. And we, to my knowledge, I have not seen other people put out their inter-rater and inter-rater analyses for driver detection. So the fact that we're doing it, I think, means that we're getting much better scores than other people have tried this. And we found, honestly, what I consider to be very good Cohen's, kappas, and inter-rater agreement. So I think we are successfully eliminating a lot of the noise and variability of the problem of the process in at least driver detection. When it comes to LG-MRI fibrosis, I know that there is a whole corpus of literature on how do you pick that correctly. And we didn't really explore that here. So I know it can be very sensitive to the thresholds that you choose. Thank you.
So in the AIM2 work, when you're doing the SimClear, your unlabeled data set, that doesn't include your labeled samples, or are they also part of the self-supervised part?
They are not included in the self-supervised learning.
So then the self-supervised, is it acting on similar data, or like, I assume it's still Orion basket data in AFib?
Yes, and it's from completely different subjects even, from the driver detection data. So we did our best to prevent any crossover between those data sets.
And then if you were, so you mentioned time clipping as another augmentation technique. What else might you think of as like your next augmentations to try that might be more relevant to signal data rather than image data?
Oh, goodness. I'm trying to think of what I had in mind. I think something like doing Fourier transforms and analysis feeding in like the spectrum data. That would be a really good next step as well. Let me think. You could look at response to, I don't know, adding QRS or not. I'd have to think about it and look at my old notes to really give you an idea. But the example I have right now is that you could do like spectral analysis and throw that in as an augmentation.
That you brought up, so this data that you use for that is this like pre-processed data with QRS subtracted and all that?
Yes.
So do you think adding that back in, because the thing people say with machine learning is like just let it figure it out, give it everything. Do you think that could help it, or would it get distracted by that big amplitude signal in the middle of everything?
I totally agree that in general, you should be throwing in the raw data if you can. In this case, I think that would be a good next step to see how it does. One of the things that SimClear does, there's two different aspects of how it works. The first is that it sees how signals respond to noise. Signals from the same mother, in theory, will respond to noises added in the same way, right? In that case, QRS, subtraction and addition, right? You would expect that a signal that had QRS added would get mapped to the same mother as that same signal without it. And you would hope that SimClear makes the signal invariant, or it makes the network invariant to that type of noise. So in theory, it should be able to ignore QRS artifacts. And then the second aspect in which SimClear works is by highlighting important regions of data. That's where the cropping and the clipping works well, is that it, in theory, if you crop onto an important section of the data, then it will know that it definitively came from a certain mother. Thank you.
Once the network is trained,
Yeah.
and what is the computational expense of running that classifier on new data? Can it be some real time perhaps? Has it to be done offline?
This is using, we're not using supercomputers here. We're using very simple GPUs, which run this in milliseconds. So this could be implemented into like a clinical process, if assuming you can get it into the mapping software, right, which is its own challenge. It could be run in real time, very, very quickly.
Okay.
Thank you.
Wondering, what's the clinical difference between focal and rotational drivers? Because I mean, when you identify, you separate them, but I just wonder if that translates into a different approach of ablation or something, or it's just how they are, how you see them.
No, great question. As it stands, the clinical approach is if you see a driver, at least in the methods that have been tried, you burn it. And as you can see, that won't be reliant on whether it was a focal ectopy or a rotor. Now, there's sort of discussion as to whether you can distinguish really between a focal ectopy and a rotor because of the way that intramural conduction works, right? If you have a ectopy on the epicardial surface, it might come through on the endocardium in a way that looks like a rotor, and vice versa for a rotor and a focal ectopy. So it's not clear that you would treat them in a different way.
Any other questions?
What motivates the use of the canine model versus other animal models for studying AFib?
Oh, that's a great question. For one, like the other contrasting models, I would probably look at are porcine and hircine models. Hircine being the adjective for goat. The big differences are that one, the conduction system is a little different, generally in the ventricles, between the ungulates. That's what pigs and goats are, whereas canines are not. They have paws. And then the canine has a more similar conduction system to humans, right? And then additionally, goats and pigs do not respond very well to serial mapping studies. You really wouldn't want to do them more than like 1 or 2 studies, just because they can be really sensitive. They have a lot more clotting factors, for example, which can make, like goats have a lot more clotting factors, which can make surgery dangerous and difficult for them.
Thank you.
Yep.
Excellent. Nice work, Bram. You're on.